Hi all, we are super excited today to be here with you to announce the launch of SYMPHONYAge, the newest generation of epigenetic clocks developed at Yale and now brought to you by TruDiagnostic. First, introductions to the team. I'm Dr. Albert Higgins-Chen. I'm an assistant professor in the departments of psychiatry and pathology at Yale. I'm a practicing physician and clinical advisor on this project. And this is Raghav Sehgal.
Everyone, I'm Raghav. I'm a computational biologist and senior member of the Higgins-Chen lab. I'm the first author of SYMPHONYAge publication and trained SYMPHONYAge and wrote the code for it. I'm also the director of bioinformatics at a longevity clinic and a co-founding employee of a Series A funded computational biology startup. Over to Albert.
So Raghav and I have been working closely with the TruDiagnostic team behind the scenes to bring SYMPHONYAge to you. Also central to our team is Dr. Morgan Levine, who, of course, is one of the biggest names in the epigenetic clock field, a fantastic scientist and mentor to both me and Raghav, formerly at Yale, now at Altos Labs. She developed the initial idea for this epigenetic clock and is a primary scientific advisor. We'll discuss today the major innovations of SYMPHONYAge. You can find all the details here in our publication. Of note, SYSTEMS-Age is the name for the general technology and framework, whereas SYMPHONYAge is the name for TruDiagnostic's implementation of SYSTEMS-Age, which you will see in our reports. As you well know, there are many epigenetic clocks. We won't go into the history or details of these clocks, since that is already covered extensively elsewhere, including in other webinars by TruDiagnostic. But I do want to share the analogy that has helped inform our thinking in developing SYMPHONYAge. And that is the traditional hourglass that measures the passage of time. This unifies the major clocks developed up to this point. There are 3 generations of epigenetic clocks corresponding to 3 parts of the hourglass. So the first generation clocks here are the sand at the bottom. They simply tell you how much time has passed since birth. This includes the famous Horvath multi-tissue clock and the Hannum clock, trained to predict chronological age. The second generation clocks here are the sand at the top. They tell you how much time you have left because they are trained to predict mortality risk. This includes PhenoAge, developed by Morgan Levine on our team, as well as GrimAge. And finally, Union and Pace, the flow of sand from top to bottom, in that it was trained to predict the pace of aging. Of note, OmicMage from TruDiagnostic was trained similarly to a second generation clock in that it predicts mortality, though with way more insights derived from multi-omic and clinical data. So really, all these clocks are giving you a different view of the same unifying picture. But before we talk about how we might improve upon this framework, it is important to remind ourselves of the promise of epigenetic clocks. So epigenetic clocks were first developed just over 10 years ago, so it is not surprising at all that it has not made its way into mainstream clinical medicine. As a physician, I first got excited about this area of research because in medicine, we don't explicitly quantify aging. We might just get a general sense of how our patients are aging. So when I'm telling another physician about a patient, I would go over loads of information about history of present illness, ongoing clinical problems, medical history, medications, allergies, physical exams, labs, imaging, etc. But aging, well, really the info is largely limited to this is a 38-year-old woman, a statement about what comorbidities they might have, and at one point when reporting physical exam, I might comment about a patient appearing older than stated age. Both of these do indicate to me that the patient is at higher risk of mortality in the future than might be expected for the average 38-year-old. But there's no real explicit quantification of aging. And indeed, increases or decreases in epigenetic age, termed epigenetic age deviation, has been linked to many different risk factors for aging and many different aging outcomes. There's now numerous publications on this. However, let's take a step back and think about what is implied by this hourglass analogy. This treats aging as a singular process that can be boiled down to one dimension, a single flow of time, straight line, single axis of aging. You get a single biological age number. Sorry about that. You get a single biological age number or a single pace of aging. Even if you get multiple numbers, it is all part of the same single dimension. And this one dimension is supposed to predict your risk of age-related functional decline, morbidity, and mortality. But remember, aging is a huge risk factor for numerous diseases simultaneously: cancer, heart disease, COPD, stroke, dementia, diabetes. The list goes on. So really, we are trying to get this single dimension of aging to predict all of these different diseases all at once. And is a single number really up to the task? Reality, different people are, well, different. We have different risk factors and develop different sets of age-related conditions. And a single biological age cannot capture this individualized risk. So SYMPHONYAge was developed to define multiple biological ages. But how do you do that? Our insight is that there is already a very natural way to define multiple ages, and that is by taking into account physiology and the fundamental organization of the human body. Human body is divided into organ systems working together. Clinical medicine is already organized by organ system. Specialties are cardiology, gastroenterology, neurology, etc. There's some drawbacks to this division for sure. We don't want to focus myopically on just one system. And we want to make sure we are treating our patients holistically and as whole persons. But there are also very good reasons to focus on organ systems. Each organ system is defined by functions that are essential for life. The cardiovascular system transports the oxygen and nutrients to keep us alive and removes waste. The kidney excretes the waste and regulates body fluid composition. The immune system fights infections. Of course, these systems are highly integrated, and organs play roles in multiple systems. But importantly, these are the essential functions we are trying to preserve in the face of aging. Each organ system can also be linked to specific diseases: dementia for brain, osteoporosis for skeletal, myocardial infarction for cardiovascular. Of course, diseases affect multiple systems, given that they all interact, but you can typically pinpoint which organ system is most involved. Then, there is already a rich suite of existing biomarkers to assess the function of each organ system. Again, biomarkers can involve multiple systems, like traditional liver enzymes like AST and ALT are also made by muscle, heart, and kidneys. But there is still relative specificity. I'm not going to use liver enzymes as the first test to evaluate your skin. And most importantly, these biomarkers are actionable. We order tests because we have some clinical suspicion of a problem, and the biomarker can give us critical information to guide next steps. They help guide diagnosis, further testing if needed, assessing severity and prognosis to make decisions like, do I need to hospitalize a patient, selecting treatments, and monitoring and prevention of complications. Example, if someone has symptoms or risk factors for diabetes, we can get hemoglobin A1C and glucose tests. And if consistent with diabetes, I will be sure to screen for common comorbidities, assesses the severity, and select appropriate treatment based on the known pathophysiology of diabetes to lower glucose and modify risk factors. And I'll be sure to get regular screening tests like eye exams, foot exams, urinary albumin and creatinine to screen for complications of diabetes. Start other medications like statins to prevent the diseases that diabetes will put you at risk for. And all this to say, these biomarkers like hemoglobin A1C are actionable, which is why we measure them in the first place. Now, this may all seem obvious, but it's important to note that this is missing from current epigenetic clocks. Organ systems tell us about the specific functions we want to preserve, the specific diseases we want to prevent, and have specific actionable biomarkers. Existing epigenetic clocks, by virtue of reporting a single number, do not tell us what specific diseases we as individuals are at risk for. And existing epigenetic clocks don't suggest any specific actions we should take. On the flip side, existing epigenetic clocks do quantify aging, and they capture the underlying molecular hallmarks of aging, which are traditional clinical biomarkers currently cannot do. And clocks can do this with a single blood test, which has many practical advantages. So, well, why not combine them? So we set out to do this with SYMPHONYAge. We leveraged extensive biomarker data from the Health and Retirement Study, collecting 133 different clinical chemistry and functional biomarkers. Because we have so many, we can actually divide them into groups based on organ systems. And so we categorize them into 11 different systems via prior clinical knowledge and additional searches. So the kidney group, for example, has adenine, cystatin C, albumin, BUN, and electrolytes. We then utilized this biomarker data together with DNA methylation and mortality data to build new system-specific clocks. So this involved a two-step process combining information from two different studies, which is very similar to how the original PhenoAge was trained. Except now we can do this for each system individually, one at a time. So we can take the lung biomarkers and use them to train a DNA methylation clock that predicts mortality. We do the same for all the blood biomarkers, completely separate from lung, and again for brain, heart, and each system individually. And this produces SYMPHONYAge. So in a symphony orchestra, many instruments must play together in concert. If you have a single clock, you can only tell if the entire orchestra is playing too quickly. But there are many parts of the orchestra. If you have many clocks, you can pinpoint who in the orchestra is playing too quickly and focus your attention on getting them back in sync with everyone else. Now, Raghav will take over to explain the detailed training and validation of systems age.
Thank you, Albert, for that insightful introduction to SYMPHONYAge. I'm now going to dive a little deeper into how the system scores in SYMPHONYAge have been trained. To start with, let's briefly talk about the number of biomarkers and samples that went into training system-specific scores in SYMPHONYAge. As Albert mentioned, we use a total of 133 biomarkers distributed across 11 systems. These 133 biomarkers can be found in almost 10,000 samples of the health and retirement study, and were thus used for training purposes. Additionally, we use two DNA methylation cohorts for training: the health and retirement study with 4,000 plus DNA methylation samples, and the Framingham Heart study with almost another 4,000 DNA methylation samples. Compare these to the number of samples that used in training for some of the other state-of-the-art clocks like GrimAge and Dunedin Pace, and you would really start to see the benefits of our training paradigm. We use a much larger and diverse number of samples for training system scores, which eventually lead to some very exciting results. And with that, let me talk a little bit more about the actual training pipeline for these system scores. So to build a system-specific score, we follow 3 steps. In the first step, we combine the biomarkers into unified components. Take, for example, the heart system. We take the 7 different map biomarkers like heart rate, blood pressure, BMI, and more, and reduce them into key heart components. In step 2, we predict these key heart components using whole blood DNA methylation. This gives us DNA methylation proxies of heart components. We no longer need the original biomarkers or the heart components, just their DNA methylation proxies. In step 3, we combine these DNA methylation proxies for the heart components into a single mortality predictor. The score we get from this mortality predictor is what we call the heart score. We repeat these 3 steps for all the 11 systems. Eventually, we combine the 11 system scores into one unified mortality predictor for whole body aging as well. This totals up to 12 unique system scores in SYMPHONYAge that quantify system-specific as well as a unified whole body aging score. As you might expect, this unique paradigm of training DNA methylation aging biomarkers from a systems perspective gives SYMPHONYAge many advantages over clinical and functional biomarkers. To start with, SYMPHONYAge is a single blood test that can measure aging in multiple different systems of an individual's body. The fact that it is a single blood test allows for easy and repeated measurements. This in turn enables standardization. Secondly, clinical biomarkers may vary both in type and measurement technique across cohorts, making them difficult to compare. On the other hand, SYMPHONYAge is based on methylation sites that are consistently measured across diverse cohorts, allowing for simplified comparison and validation. Additionally, SYMPHONYAge has been trained using DNA methylation features that offer high technical reliability. This is critical for assessing changes in longitudinal studies, such as those studying the effects of interventions. We will talk about this more in our webinars toward the end as well. Last but not the least, SYMPHONYAge is the first system-specific biomarker panel which is trained to predict mortality risk and has been validated across multiple large cohort studies. On that note, I would like to briefly highlight some stats on the different cohorts SYMPHONYAge has been validated in. SYMPHONYAge has been validated in 6 different cohort studies, such as the Baltimore Longitudinal Study of Aging, Swedish Adoption Twin Study of Aging, and more. Across these 6 cohorts, we had upwards of 8,100 DNA methylation samples on which we calculated SYMPHONYAge scores along with some other state-of-the-art clocks. And we associated these clocks with 28 different aging phenotypes. I wanted to state these numbers just to give you a sense of how extensively we have validated SYMPHONYAge. And with that, let me actually show you some results of our really exciting validation. One way to think about validation of SYMPHONYAge is to see if the scores are specific. That is, if they should be able to predict age-related changes in systems with high specificity. So let's see if the scores are indeed specific. So to understand the forthcoming figures a little better, I will need to explain a bit more of the plot on the right and how they have been summarized visually on the left. To start with, on the right of the screen, you can see a forest plot. This forest plot represents a cohort and race-stratified meta-analysis associating a particular system score to an aging phenotype. In the example shown in the slide, we are associating the heart score with time to coronary heart disease events. We perform this association analysis for all the different scores and record the z-score of association with the aging trait, in this case, future CHD events, for each system score. We then scale the z-scores from largest to smallest to visually represent on the body plot on the left, such that the system score, which is most associated with an aging phenotype, has the largest text size. In our example here, we can see that after scaling the z-scores, it shows that it is the heart score which is the most associated with future CHD events. This is a great validation of the specificity of the heart score. Now let's look at a few more such examples. To start with, let's look at some functional phenotypes. When we look at associations with cognitive function, we see it is the brain score that is most associated. With physical function, it is the musculoskeletal score which shows the most association. And when looking at total comorbidities, it is the whole body inflammation which shows the most association, as one would expect. When looking at future disease events, in these cohorts, there was a 7-year follow-up. That is, whole blood DNA methylation was collected in 2014, and these individuals were followed up till 2021 to see for if there were any different disease events that were occurring. Given this data, was collected in 2014, we can check if our scores were predictive of any future disease events. And turns out they are. And that too with very high specificity again. For example, the lung score is most associated with lung cancer events, or the blood score is most associated with future leukemia events, or the hormone score is most associated with future breast cancer events. And as we had previously shown, the heart score is most associated with future CHD events. We also try to see if the scores are associated with baseline disease events. We observed that diabetes was interestingly associated with musculoskeletal, but additionally metabolic and inflammation scores. Hormone was most associated with thyroid disease, and the musculoskeletal score was most associated with arthritis. We also want to highlight, we saw very replicable results of similar or same aging phenotypes in different cohort studies. For example, both in WHI and BLSA cohorts, when associating diabetes, the top 3 scores were musculoskeletal, inflammation, and metabolic scores. Similarly, when looking at CAD and CHT in BLSA and WHI, the top 2 scores were heart and inflammation. Another example we saw was when looking at cognitive function and whole brain volume in WHI and ADNI, the most associated score was the brain score. All of these just go on to show that these scores are very much replicable with their specificity. It is also very important to note that this specificity does not come at the expense of predictive capabilities when compared to whole body clocks. When looking at a multitude of conditions, we observe that the most appropriate system score predicts aging equally and sometimes better than the whole body aging clocks, while providing something the whole body clocks do not provide, that is insight into the systems themselves. With this, I hope I have been able to convince you all that to some extent that SYMPHONYAge scores are both specific and equally, if not more associated to aging phenotypes than the state-of-the-art clocks that are out there. And now, till now, we have been talking of system scores independent of each other, as if they exist in silos in the body. But in reality, that is not the case at all. These systems interact with each other and are dependent on each other. But since these systems are interlinked, it also means that these systems fail. And when they fail, they fall like dominoes, where one system may put strain on another system when it starts to fail or fall. Now, just like dominoes, these systems too can arrange and fall in different patterns. And we call these different patterns of falling of the systems aging subtypes. Let's dig a little deeper on how SYMPHONYAge can be used to understand more about these aging subtypes. As I mentioned, systems are inherently related. We can see system scores like heart and lung being highly correlated, pointing at a cardiopulmonary axis, or metabolic and inflammation being highly correlated, pointing at metaflation caused due to obesity. All of this points to the existence of aging subtypes potentially because systems are correlated to each other, but it wasn't proof enough for us. The biggest anecdotal proof we got was when we looked at three individuals who had the same chronological age and very similar whole body biological age, but they had huge differences in their system-specific scores. This really highlighted the existence of aging subtypes. What we really needed to see now was that did such subtypes exist in larger cohort studies, and do these subtypes have different disease occurrences? Turns out, when we clustered 2,100 plus individuals in one of our cohort studies based on their system scores, we observed 9 different clusters. Each of these clusters seem to have very different average age accelerated system specific scores as shown here. But most interestingly, these clusters had very different risk of developing diseases or having certain phenotypes. The heat map here shows p-values of increased occurrence in red or decreased occurrence in green with p-values listed on the cell itself. But let's focus on a few clusters to understand this analysis better. I call these 3 clusters the smoking aging subtypes. All 3 of these groups have an increased number of smokers in them, which may explain the accelerated heart and lung aging across all 3 of them. But what is interesting is that only cluster 8 and cluster 9 develop lung cancer. And out of the 3 clusters, only cluster 9 goes on to develop CHD or coronary heart disease. These 3 clusters, even though have the same common habit of smoking, which may cause accelerated aging, the effect it seems to have on them seems to be very different, and potentially something one could not have figured had we not been looking at these different aging subtypes. We just talked about cluster 9 developing CHD, but there is another cluster that develops cardiovascular related diseases, which is cluster 3. Now, both of these clusters, as you can see, have very different aging patterns. And interestingly, cluster 3 has an underrepresentation of smokers. Cardiovascular events in cluster 9 seem to be driven by accelerated heart and lung scores, as we just talked about, while that in cluster 3 might be the effect of accelerated heart, metabolic and inflammatory aging. Now imagine individuals from these 2 groups coming to a physician for cardiovascular concerns. They may be treated very similarly, but given their aging subtypes and distinct aging in their systems, maybe, just maybe, they should be treated very differently. And asking and answering questions like these is what really the aging subtype analysis using SYMPHONYAge allows us to do. In my last section, I would like to talk about quantifying the effects of anti-aging interventions using DNA methylation biomarkers. I specifically want to highlight how whole body clocks may fall short on providing us insights on anti-aging interventions, and how SYMPHONYAge, a system-specific biomarker, can fill this gap. As Albert discussed in the start, most state-of-the-art clocks measure whole body aging. Now, this is great if an intervention is affecting whole body aging, in which case the clock will be able to capture this change. But in many scenarios, interventions have very specific effects on systems, in which case a whole body clock may not be able to measure this change very effectively. Thus, we may require system-specific clocks to measure such changes. We wanted to test this hypothesis. So we looked at a few longitudinal studies where an anti-aging intervention, for example, metformin, was given for a fixed period of time to individuals, and their whole blood DNA methylation was collected pre and post the intervention. This way, we could calculate age-accelerated clock scores for each individual and do a paired t-test to see if the scores were changing with the intervention or not. Now, we looked at 3 interventions: smoking cessation, gastric bypass, and metformin. We looked at 10 different state-of-the-art clocks, ranging from PhenoAge to PrimAge to Dunedin pace. Of the 30 different tests we did, only one clock, which is PCPhenoAge, seemed to have gone down with metformin. No other clocks seem to have decreased with these interventions. Based on this data, one might even say that none of these interventions had an effect on DNA methylation aging biomarkers. But maybe, just maybe, we weren't looking at the right DNA methylation aging biomarkers. As we look at SYMPHONYAge scores, we see a very different picture altogether. For example, with smoking cessation, we see a bunch of clocks going down, and specifically the lung score is the one which shows the most decrease in after having this intervention. With gastric bypass, it is only the metabolic score which seems to be showing a decrease in this biomarker. With metformin, many clock scores go down, but it is only inflammation and metabolic scores that seem to be leading the charge with the most reduction. All of these examples just highlight the power of using system-specific scores over whole body scores when looking at anti-aging interventions. We are performing this study for over 50 anti-aging interventions and 100 plus DNA methylation biomarkers, all to understand how anti-aging interventions affect DNA methylation biomarkers. We will be talking more about this in our future webinars. And with this, I will hand it back to Albert to talk more.
Okay, thank you, Raghav. Raghav just showed you the evidence that SYMPHONYAge is specific as a predictive biomarker. Aging in specific systems will predict specific aging and health outcomes. And as Raghav hinted, we are actively investigating whether SYMPHONYAge is specific as a response biomarker, that anti-aging interventions will affect specific systems. But I want to make clear, it is still an open question whether any of these clocks can be used as surrogates, where the change in the epigenetic clocks due to interventions is meaningful for outcomes. That would require a much higher level of evidence, and that it has to be an active area of investigation for the geroscience field as a whole. But could SYMPHONYAge be clinically actionable in other ways? So clinically actionable means that the biomarker can guide what clinical actions we take to benefit patients. We need clinical experience and far more studies around this. And that is why we want to know how you will use SYMPHONYAge. We currently use chronological age for a wide variety of reasons in the clinic, in our diagnostic process, in assessing risk for various diseases, anticipation of medication side effects, preventative medicine guidelines, and so on. Well, what if we used SYMPHONYAge instead? What if we gave shingles and pneumococcal vaccines based on immune age instead of chronological age? What if we avoided anticholinergic medications in individuals with high brain age? What if we used lung age to determine when to screen smokers for cancer? And to be clear, 100% clear, we have no current studies to support this. But that is why we put this out to you. How will you use SYMPHONYAge? And how can we design new studies to determine the role of epigenetic clocks in the clinic? And with the rich information provided by SYMPHONYAge, we anticipate you'll find applications we haven't thought of. We're all in this together. We're working as a community to discover clinical applications to improve human health in the face of aging. We'd like to thank the whole team at TruDiagnostic, and we thank all of you for listening and trying out SYMPHONYAge. Thank you.